So in 1996, an important discovery was made. By blocking a signal on white blood cells, Professor Jim Allison was able to activate the immune system and cure mice from cancer. This was a starting point of the greatest breakthrough in cancer therapy of our time, immune oncology. Also highlighted last week by the Nobel Prize for Professor Allison and Professor Honyo. It's extremely successful. It will save millions of lives. Still, in the end, about 1 out of 3 people in the Western world would die from cancer, unless therapy improves further. And that's what Alligator is here to do. We develop the next generation immuno-oncology antibodies, tumor-directed immunotherapies. So what is tumor-directed immunotherapy? Basics are the same as for basically all other immuno-oncology companies. We develop therapeutic antibodies, they activate the immune system, and they are directed towards validated immune pathways. The difference is that our compounds are all engineered to have a stronger activity in the tumor area versus the rest of the body. The whole point is to focus the immune attack towards the tumors and reduce systemic side effects. Here, Alligators, we are pioneers. We have 4 products today in preclinical and clinical development, all with the first and best-in-class potential. Our business model is to develop antibodies from ID generation up to clinical phase and followed by out-licensing to pharma, international pharma. What's significant about Alligator is that we have already validated both our pipeline and our business model through the major partnership with Johnson & Johnson. It's a $700 million deal plus royalties on all future sales. One of the greatest deals made in this sector in Scandinavia. Based on the upfront payments and also successful share issue at our IPO, we have also a very stable financial situation with money in the bank for the next 2 years. Our success factors are twofold. One is a very strong technology platform. That was founded Alligator 16 years ago. We have a human antibody library. This allows us to select therapeutic antibodies towards any conceivable target. We have a protein evolution technology, allowing us to optimize any property of those antibodies. And we also have a bispecific platform. We can generate superior compounds by actually fusing 2 different therapeutic antibodies into one superior molecule, creating new biology. And that's our bispecific platform. We have 2 different formats, and they are acting in concert with our antibody library. Plus, we have added a CMC capability. We are today able to manufacture our own antibodies up to the stage of GMP manufacturing, where we outsource. The other success factor is our R&D environment. Of the 50 people at Alligator, 43 are within R&D. 2/3 have a PhD, which is 28 researchers with a PhD in our company, all with a long experience in protein engineering and immuno-oncology. We started out, founded in 2001, on protein evolution technology. In 2008, we entered the field of immuno-oncology, and that's highly significant. It's 5 years before this field was generally accepted. That's why we today stay with a pipeline with international standards and very competitive. In 2015, 1013 entered clinical phase 1 and was out-licensed to Johnson & Johnson, and is now run by the Johnson & Johnson company Janssen in clinical phase 1. Next year, the next product, a bispecific product, entered preclinical development, and then last year, 2 more products in preclinical development. And now our bispecific CTLA-4 product is transitioning to clinical phase as we speak. So what is tumor-directed immunotherapy? What's approved today is on the left side. General immune activators. They activate the full immune system. It's extremely effective. The one problem is that the immune system does attack not only tumors, but will also start to attack healthy tissues, creating a lot of quite severe side effects. What we try to do is selective activation of the relevant part of the immune system. The part of the immune system that's in the tumors, in the metastasis. We have a number of different concepts to achieve this. One significant concept is to generate bispecific antibodies. We can fuse a tumor-targeting antibody with an immune-activating antibody and fuse them into one molecule. Such a molecule can be injected in the circulation, it will distribute, and then it will accumulate in the tumors through the tumor-targeting antibody. But we have actually made one more significant advancement here. They can be engineered so they are inactive while circulating. So we can actually inject inactive antibodies. Circulating through the body, and then they accumulate in the tumor, and only once they are at the tumor, they will become activated through the binding to the tumor cell. So there is competition. Today, there is approximately 1,000 products claiming to be immuno-oncology antibodies. If we look at the core of immuno-oncology, which is antibodies that induce long-term immunological memory, then we're down to around 250 products, still quite a large number. On the market, it's PD-1 and CTLA-4. Same goes for phase 3, and then we have a number of other targets also in phase 2, 1, and preclinical development. If we look at bispecific antibodies only, we're down to around 60 products, mainly in preclinical phase. This is still an emerging technology. And if we go even further and only look at bispecific tumor-directed antibodies, we're down to about 25 products. And here, Alligator actually has a quite significant proportion. In our pipeline today, 1013 is in clinical phase 1. It's partnered with Janssen Johnson & Johnson. I'll come back to this product. 1015, we're first in the world with a new concept, a tumor-localizing CTLA-4 product. It's just about entering clinical phase 1. And then we have 2 more products that are going to enter clinical phase next year. So let's go and look into the products. 1013. Here is a mechanism of action. And this is also immuno-oncology in a nutshell. There are 2 parts of the immune system that are important. On the one side is the antigen-presenting cells. They show the immune system what to attack. On the other side is the soldiers of the immune system, the T cells. They execute the attack. And immuno-oncology is the interplay between those 2 cells, the communication between those cell types. 1013 activates the antigen-presenting cells. You can look at it as a billboard. It shows what to attack. When 1013 activates, it will light up, and it will be much clearer to the soldiers what to attack. The message will be much clearer. This communication, if we look in more detail, here is the antigen, the tumor part that is recognized by the T cell. The antigen-presenting cell shows the tumor part. It is recognized by the T cell, and that will send a signal to the T cell. That signal says, this does not belong here, should be attacked. And that will trigger the T cells. But the T cell asks a control question. Is it dangerous or is it not dangerous? And that's where all the other signals come into play. The red signals, you can recognize PD-1, CTLA-4, the Nobel Prize winners. They both send the message, it's not dangerous. So the T cell will receive, does not belong here, but well, it's not dangerous, so we can let it be. That will turn off the attack. That's the kind of signal the tumor will use to escape the immune system. Those signals we must block. That's why we have PD-1 blockers, CTLA-4 blockers on the market today. The other signals, they send the signal, it's dangerous, and that will trigger an immediate attack. Those must be activated by stimulatory antibodies. CD40 is one such, but you can see it's the only signal that's specific for antigen presentation. And that makes actually CD40 quite a unique target. It can be combined with any T-cell activator, including PD-1. It's also ideal for example, combination with cancer vaccines. A cancer vaccine is to provide the cancer antigen. CD40 improves presentation of that antigen. And that's one of the plans going forward. Where we are today, we have completed at Alligator one clinical trial that was intratumoral dose escalation. That demonstrated that it's safe and tolerated at clinically relevant doses. Johnson & Johnson, or Janssen, they have almost completed another dose escalation trial. It's intravenous dose escalation, and it's a very large trial. Together, it's more than 100 patients in those two trials. And we are now about to select a dose and then start combination with PD-1. All further clinical development is by Janssen, and they also take all costs. So it's a quite good deal. Deal structure in more detail, we have a close to 700 million milestone arrangement. We have received 46 million US dollars today. And it is a pretty front-loaded arrangement. More than half of those milestones is actually on development and registration milestones. And then the second half for sales-based milestones. Plus, we have royalties on all future sales in the range of 10%. Competitors, yes, there are coming more and more competitors. It's a hot target. I believe the key competitor is Apexigen. They are slightly ahead of us now. They have a number of combination trials already initiated. Roche and Seattle Genetics, they have both tolerability issues, and Celldex and AbbVie are a few years behind. So I think we are at the front. We have a very strong partner, and we also have a best-in-class potential with our products, which looks pretty good. The good thing is that Janssen takes all the costs, and that means we can focus on 1015 and the next projects. So 1015 is a first-in-class product. Here, we provide a combination therapy in one molecule. 2 immune-activating pathways are activated at the same time, OX40 and CTLA4. And it's also an interesting product from an engineering perspective. The OX40 antibody has been selected through our phage display library, awarded Nobel Prize last week by Arnold. Sorry, by Winter and Smith. We have also optimized the CTLA4 binder through our protein evolution technology, also receiving Nobel Prize last Thursday. And that was Arnold. And then optimized through Borrebeck. And then we have fused those through a bispecific format. And the third Nobel Prize is a target, CTLA4. So there's actually 3 Nobel Prize in the same compound, which is quite a coincidence. We hope with this product not only to be as good as a combination of an OX40 antibody plus a CTLA4 antibody, but actually to be superior. And why is that? Well, we can activate both pathways, but we can also bring cells together by forming a physical bridge between those two receptors. And that has been clearly demonstrated now. Here is an assay where we look at cell-to-cell crosstalk interactions. Basically, if you mix OX40 and CTLA4 antibodies, there is no effect. With the bispecific molecule, we can adhere the cells together, and that will increase cell-to-cell immune crosstalk. The important thing, though, is that this is associated with superior efficacy. Here is an assay, the key mechanism of action, which is killing suppressive T cells that will allow activation of the T effector cells. And just to make it short, the blue line is a combination of both OX40 and CTLA4 antibodies at the same time at increased doses. 1015, though, has about 3- to 4-fold better effect than those two together. And this translates to actual anti-tumor activity as well. Here's a model, it's a bladder cancer model. We have 4 different tumor models, all demonstrating activity. In this model, we can cure around 65% of the mice from bladder cancer. And that is impressive. But the most impressive part of this, once the mice are cured, they are immune to that cancer. And that's why patients keep live for a long time. They give long-term survival because they become immune to the cancer. And this is also demonstrated here. If we take these survivors and then wait a few months, no further treatment, they will 100% be immune. If we induce another tumor that will grow, naive tumors, not exposed, will grow, but once they are cured by 1015, they will be immune for the foreseeable future. So then the question is, does this translate to patients? Well, we know it does. CTLA-4 is extremely well validated, hence the Nobel Prize, I would say. We know its clinical efficacy in patients. The key problem today is the toxicity. CTLA-4 is effective, but it's very toxic. And we have a beautiful solution to that by localizing our product to the tumors. And that's why our next slide is so important and so exciting. What we've done here is to look at the immune activation in the tumors. Non-treated tumors, very low immune activation. You can improve that by CTLA-4 or OX40. 1015, though, is several-fold more efficient. But the really good thing about this is that this occurs in the tumor. In other parts of the body, there is no effect whatsoever. So potentially, we can activate the immune system through CTLA-4 in the tumor without doing so in the rest of the body, thus avoiding systemic toxicity. We are about to enter a clinical trial. We have submitted the clinical trial application. We are waiting for regulatory approval, and we will open up the study for recruitment later this year. It's a traditional phase 1 trial of up to 50 patients. We will try to identify a safe and tolerable dose. And that's what's so important here, because we know efficacy with the target. If we can demonstrate tolerability, we have a great product. I'll come back to that. On the financial situation, very short, it's a good situation today. 500 million in the bank. That will take Alligator for about 2 years with the current pipeline without any of the expected milestones from the products. So in summary, 1013 is in clinical phase 1. We have a strong partner. We have a very great target with a unique profile, potential to be best in class, and we are at the front. 1015, we are first in the world with a new concept, a tumor-localizing CTLA-4 bispecific product. Let me just highlight the potential of this product. There are today only 2 targets in immuno-oncology demonstrating clear clinical efficacy, PD-1 and CTLA-4, hence a Nobel Prize for both targets. There is one key difference. PD-1 sells for around $10 billion last year. It will increase to around $30–40 billion within the next 5 years. It's an enormous market. CTLA-4 sold for a little bit more than $1 billion last year. It's quite a lot, but it's clearly much less. And it's expected to increase to maybe $2 billion in the next 5 years. So why the difference? The key difference is toxicity. Patients don't take CTLA-4 even if they know they have a much better chance to survive. And here, 1015 provides a beautiful solution. We can localize the antibody towards the tumors. If this can be confirmed in the next clinical trial, we have a huge product. So just to conclude, value triggers, 1015 is entering clinical phase 1 now. Next year, we hope to get a clinical phase 2 milestone, and that will be by the end of next year. And also, we have 2 more clinical entries. 1017 will start. We will submit the CTA mid next year and start a trial in the autumn. And for 527, we will submit the CTA end of next year and start a recruitment early 2020. So by that, we are establishing a very solid clinical pipeline with 4 products in clinical phase by the end of next year. And then we show that Alligator is becoming a key player within our niche of immuno-oncology, which is tumor-directed immunotherapy. Thank you.